Welcome to this explainer. Let's jump right into something that's honestly straight out of a botanical crime scene investigation. Today, we're opening up the fascinating detective toolkit that scientists use to identify the powerful and sometimes incredibly deadly chemicals hiding inside plants. You might be wondering how researchers actually figure out what's inside a leaf, a root, or a piece of bark. I mean, you can't just look at a plant and magically see its medicinal compounds, right? Instead, you have to force the plant to reveal its secrets. And they do this using simple chemical reactions that act exactly like a fingerprint. Basically, by introducing very specific chemicals to a plant sample, it triggers these unique, visible reactions that tell us exactly what's hiding inside those plant cells. To keep this explainer organized and easy to follow, we'll go through the evidence step by step. We're going to cover cracking the plant code, identifying potent alkaloids, spotting colorful glycosides, testing for tannins, exploring terpenoids, resins, and oils, and then finally reaching our chemical verdict. Okay, let's start right at the beginning with Section 1, Cracking the Plant Code, where we test the foundational structures of the plant itself. Before we get into the specific tests, let's just quickly define our core tool here. Pharmacognostic tests are essentially chemical interrogations. You take a drug substance derived from a plant, and you apply a specific reagent to it. The crucial point is that we're looking for fireworks in a test tube. We want dramatic, undeniable visual proof, like a sudden color change, a strange smell, or a physical transformation, that confirms exactly what chemical we're dealing with. For example, if we want to know if a plant part is packed with energy-storing starch, we just drop a little dilute iodine solution onto it. Boom, it instantly yields this vivid, unmistakable blue. On the flip side, if we're testing for structural components like lignin, we use the fluoroglucinol test with some hydrochloric acid, and it bursts into this bright pinkish red hue. Interestingly, we see that exact same pinkish red when we test for mucilage and cellulose using ruthenium red. It's incredibly straightforward and highly visual. Which brings us to our first major class of chemical suspects in Section 2, identifying potent alkaloids. Now, this is a family containing some of the most potent, intense, and frequently dangerous drugs known to humanity. What's really fascinating here is that we can use a suite of general tests just to confirm if an alkaloid is present at all, long before we figure out which specific one it is. Instead of bright, clear colors, these general tests create what we call a precipitate, basically a cloudy, solid substance that forms in the liquid. Mayer's test using potassium mercuric iodide gives us a cream-colored precipitate. Dragendorf's test with potassium bismuth iodide drops out an orange-brown cloud. Wagner's test using iodine and potassium iodate gives a reddish-brown. And Hager's test, which uses the sodium salt of picric acid, yields a yellow precipitate. It's an amazing preliminary lineup to just confirm, yes, an alkaloid is in the room. But sometimes these preliminary results are way more dramatic. Take tubacororine, for instance. It's a famous muscle relaxant that was historically used on poison darts. If we hit that with mercurious nitrate, the result isn't just some dull, cloudy liquid. It's a shocking, vibrant, cherry red reaction. It's literally nature waving a giant red flag at us. Once we confirm we have an alkaloid, we start building specific detective profiles to narrow it down. Let's say we test for morphine using nitric acid. That turns red. And crucially, codeine doesn't do that. The deadly poison strychnine, we can single that out by applying ammonium vanadate and watching for a distinct purple reaction. Or, get this, cocaine is identified not by a color change at all. You add sulfuric acid and detect a very distinct benzoate smell. Finally, the Van Erck test rounds this out, turning blue when ergot alkaloids are present. Moving right along to Section 3, spotting colorful glycosides. These compounds are heavily used in modern medicine, showing up in everything from harsh laxatives to absolutely life-saving heart medications. Glycosides have a really wide range of behaviors. Testing for anthraquinones using Born-Trigger's test, which involves an alkali and ammonia, yields colors anywhere from pinkish red to violet. But look at the cardiac glycosides. By applying the Lieberman-Burchard test with acetic anhydride and sulfuric acid, the solution turns this vivid, undeniable green. And saponins? They are almost playful. Testing them doesn't even require any harsh acids. You literally just mix the drug with water, shake it up, and look for a visible, persistent froth formation, exactly like soap. But we must always be incredibly precise when we're dealing with medications for the heart. If you're specifically looking for the powerful cardiac drug digitalis, Hitting it with the legal test, which uses pyridine and sodium nitroprusside, will trigger a really striking red color. That is an absolutely essential test for a botanical compound where the dosage margin between healing someone and harming them is razor thin. Now let's dive into our fourth suspect group, Section 4, testing for tannins. If you've ever had a cup of black tea that made your mouth feel kind of dry and puckered, you've experienced astringent tannins firsthand. Tannins interact really heavily with metals, and their tests reflect that. Unlike the bright, flashy reds and yellows of our alkaloids, general tannin tests tend to yield dark, moody, almost gothic colors. The ferric colloid test gives us a deep bluish black, and ferrous sulfate yields a heavy brown black. Now, potassium ferricyanide is the outlier here, giving a deep red, but generally when you see those inky blacks and blues in the test tube, you know for sure you found tannins. What's really fascinating is how we can differentiate within the tannin family itself, because not all tannins are created equal. By using completely different chemical reagents, we can split them apart. Hydrolyzable tannins will reveal a blue color when you hit them with ferric chloride. But non-hydrolyzable tannins ignore that entirely. Instead, they show a red color when tested with sulfuric acid. It really is all about finding the exact right chemical key for the lock. For our final major chemical group in Section 5, we're looking at the aromatic, highly volatile world of terpenoids, resins, and oils. These are the compounds that give plants their incredible, distinct smells and their sticky textures. The sheer consistency of these particular reactions is striking. When you're testing for terpenoids, there really isn't much room for ambiguity. Whether you're using the Sudan test, the scarlet red test, or Tilden's test with nitrosyl chloride, terpenoids definitively announce their presence by flashing a bold, deep red. It is a highly, highly reliable signal for this specific class of chemicals. So the ultimate point of all these tests isn't just knowing what a plant is. It's about catching fakes. The botanical trade is historically full of counterfeit materials. Take benzoin resin, for example. An ether test is all a botanical detective needs to distinguish the genuine article from a cheaper imposter. True Sumatra benzoin will predictably turn brown in the presence of ether. But if someone tries to sell you false Siam benzoin, the ether test exposes the lie instantly by turning a purple-red color. The chemistry just doesn't lie. And that brings us to Section 6, the final chemical verdict. We have analyzed the evidence, run the chemical interrogations, and systematically categorized our results. Honestly, seeing how vividly these plants react to our chemical tests, from the deep red of a terpenoid to the froth of a saponin and the cherry red of a muscle relaxant, leaves you with a real sense of awe. Every single leaf or root you walk past outside is a complex chemical factory. It really makes you wonder about the untapped botanical mysteries still waiting out there to be discovered, and the clever new tests we'll need to invent to crack their codes. Thank you so much for joining me on this journey through the Botanical Detective Toolkit. If you found this explainer helpful for decoding the incredible world of pharmacognosy, please remember to support our work. Please like, share, and subscribe our channel so we can keep bringing you clear, structured explainers just like this one. Keep learning, stay curious, and I'll catch you in the next one.